Hi everybody, I'm the greatest living inventor, and today we are doing my invention of a solution for mosquito-borne diseases. So let me write it down. Solution for mosquito-borne diseases. Okay, so I'll take off my jacket first. Okay, so this is taken from my musical about GMO and CRISPR-Cas and gene drives, and so it's part of the musical. Okay, so the musical is called Little Lab of Horrors. Okay. Little Lab of Horrors, which is a parody of Little Shop of Horrors, and all the songs are with the same melody, but melodies, all the songs, but with the different words that I wrote, and all the musical that I wrote. So it's a musical. Okay, so this whole invention is inside the musical. Two characters are talking with each other, so I left it as is. So I just copied it from my musical. Okay, so now the description of the invention. Fight the diseases that mosquitoes spread by cleaning off the mosquitoes' saliva and proboscis, the straw, with high energy beam, and by dialysis filtering that cleans the mosquitoes' blood, hemolymph, from pathogens. Okay? So I'm just writing it so you get it more constructed. Okay, so fight the diseases that mosquitoes spread by cleaning of the mosquitoes saliva and proboscis okay, this means straw. Let's write it here. Straw. With high energy beam. and by dialysis, filtering, that cleans the mosquito's blood, hemolymph, from pathogens. Okay, so this whole thing is the description, and since I want to stop as little as possible during the invention because it got the flow of the musical, then it's not music, but it's not a song, but it's like a talk between two characters, so I want you to have the whole picture first. Okay. This invention is worded / phrased as a dialogue because it's from my Little Lab of Horrors musical. Thank you. It works in an original way. Have you noticed how all these solutions are deadly in one way or another to the mosquito? So my solution is based on keeping the mosquito alive and well and only killing the disease pathogens inside it. If you think about it, when you damage the mosquito in any way, you work against evolution, because any mosquito that survives will carry the genes that allows it not to be killed by you. For example, fear of traps and so on. But if you think about it more, our problem is not with the mosquitoes. They are annoying, but what we really want to fight are almost always viruses. Yellow fever virus, dengue fever virus, Zika virus, Japanese encephalitis virus, West Nile virus, and so on. The only exception is malaria, which is a single-celled microorganism of the Plasmodium group. So what we want to do is to catch the mosquito, reward it by a drop of food or blood so it comes again next time, and in this machine that catches, it would do one of two things or both. Method 1, dialysis filtering that cleans the mosquito's blood, hemolymph, from pathogens. Okay, so this is the dialysis filtration of the blood of the mosquito, the hemolymph, from pathogens. And method 2, that was method 1. Method 2, cleaning the mosquito's saliva and proboscis, straw, okay, that part that has the mosquito has a straw that it sucks the our blood into its mouth with high energy beam. Okay. So here I'm giving you a link. I will put all the links in the description of the video. Wikipedia, sterilization, microbiology, hashtag ionizing radiation sterilization. Okay, so I'm just writing the words. Let's erase everything. I'm sorry the eraser doesn't erase. It's I have two erasers, so I clean them with soap and water and dry them, and I clean the board with some window washing material and dry it also, and nothing helps. So sorry. Maybe it's a little bit also because I changed the marker. The last one was weak, so I already. So this is a new one, so maybe that's it. I don't know. So I apologize. You've seen that this was squeaky clean before I wrote. Okay, we'll do what we can, like everything in life. So sterilization, microbiology, ionizing radiation sterilization. Ionizing radiation sterilization. Okay, so in Wikipedia you can see this. Can you see? Yeah, okay. So the way that a mosquito transmits the disease is by biting a sick person and then biting a healthy person. So if the mosquito will be cleaned between these points in time, the disease will not spread. So we want the mosquito to visit the cleaning machine as frequently as possible. That's why we never harm the mosquito, and we always give it a reward in the machine. We will not have less mosquito bites, which is the annoying part, but they will no longer spread the disease, which is the critical part. Insects have a heart, and it pumps a fluid that has the same job as blood in us. It's called hemolymph. Remember, we wrote hemolymph. So the insect's blood is called hemolymph. It's not important. I just want you to, if I'm saying hemolymph and forget to say that it's the insect's blood, that you will know what I'm talking about, or you read it somewhere. So insects have a heart, and it pumps a fluid that has the same job as blood in us. It's called hemolymph. Now a link I will put in the description. How is bug blood different from our own? By Rob DeSalle in Scientific American, March 19th, 2001. Okay, from Scientific American. I'll put all the links in the description. How is, no, sorry, I read this. So the machine will tempt the mosquito with a drop of food or blood. We can also attract the mosquito with the smell of CO2 or warmth or whatever attracts the mosquito. Okay, the way it finds us, by CO2 or warmth. But it's important also to reward it so it comes back often. Once the mosquito is inside the machine, we radiate the area of its head with X-ray or electron beam. Okay, so either X-ray or electron beam. It's not the same, either or. This messes up the DNA of the virus, so it's no longer functional. Okay, so we radiate the area of the head of the mosquito if we have the mosquito. I don't know how to draw the mosquito, but let's say it has a trunk and it has a head and it has these things like dengue mosquito, and it has wings, and this is the mosquito. Okay, so we are radiating. Let's draw the radiation in pink. Okay, so radiation. Can you see the pink? No. Okay, let's choose some other color. Okay, so radiation. And we radiate the area of the head of the mosquito. And we need to protect the other part of the mosquito, so we will not sterilize the mosquito because we want the mosquito to reproduce afterwards. We don't want to kill the mosquito. We don't want to sterilize the mosquito. We want the mosquito to be healthy, just without the parasites in here. The parasites are deadly to us. The mosquito is not deadly to us, it's just annoying. Okay. So the machine will tempt the mosquito with a drop of food or blood. Okay, let's say here is a food or blood. And the mosquito comes to eat. Okay, and we want some, this would be the machine, and we want the machine to include, so the mosquito can enter here, okay. The mosquito can enter its head, but not the rest of his body. Or even the trunk, the straw, okay, but not the rest of his body. Okay, so this messes up the DNA of. Once the mosquito is inside the machine, we radiate the area of its head with X-ray or electron beam. here in light blue and in cyan, and we radiate the area of the head with X-ray or electron beam. This messes up the DNA of the virus, so it's no longer functional. The other thing we do is equivalent to what we do with the person kidney failure. The other thing we do is equivalent to what we do with the person kidney failure, which is dialysis. Okay, so now we go to the second thing that we do in the machine, which is dialysis. Okay, if a person has bad kidneys, we do this to the person. Here we do this to the mosquito. This is a mosquito. Okay, so you can see in Wikipedia, dialysis, okay, it's the changing and cleaning of the blood. The difference, the different components of the mosquito's blood, hemolymph, have different sizes, but they are all fixed and known to us. Let's say, for example, that it has 6 materials. Okay, so look in hemolymph in Wikipedia, and we are talking about more or less 6 materials. One is water. Smallest molecule here, just oxygen and 2 hydrogens. 2 is inorganic salts. A little bigger than this, like sodium chloride or table salt. 3 lipids. And this is fats. Bigger, they are bigger, each one is bigger. Okay, contain some head and then chains of carbon and hydrogens. 4 proteins. Usually a lot bigger and more complex. 5 yellow fever virus. For example, yellow fever virus, or any virus that the mosquito transfers to us. Viruses are made from proteins and other building blocks, so they are even bigger. Okay? Then you have things which are a lot bigger, like 6 hemocyte and hemocyte. Okay, so this is by order of size. This is small, this is bigger, 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 bigger. Okay, so things that are a lot bigger, hemocyte, you can see in Wikipedia, invertebrate immune system cell. Okay, so this is in Wikipedia, hemocyte, invertebrate immune system cell. Okay, 25 minutes. Okay, so what we need is a series of chambers. Okay, so in the machine I don't have room to show it, so I'm showing it outside of the machine. But we have a series of chambers. Okay, 6. Okay, and I'm just doing it higher so you see the difference. Okay, so we have chambers separated, and let's say in the beginning, and here we have holes, like this is small holes, and this is bigger holes. And this is even bigger holes. And this is even bigger holes. And this is even bigger holes. Okay, this is all microscopic, but this is more or less what we have. A chamber, big holes, smaller holes, smaller holes, smaller holes, smaller holes, smaller holes. And we put here the blood. Okay, this is the blood. This is not our blood, so let me draw it in another color. This is the blood of the mosquito. So we put the blood here. Okay, this is the blood of the mosquito, hemolymph. Okay, the mosquito's blood, the insect's blood. And let's say we rotate this, so in a centrifuge or something, so there is like an attraction to here, like the gravity is towards me. Okay, so it tries to fall towards me. It's like we made this upside down. Okay, so the hemolymph is here, all the ingredients, and then here is the hemocyte cannot pass because it's too big for the holes, but all the other ingredients can pass. Okay, so the yellow fever and everything can pass. So here is like here there are less ingredients, and here there are less ingredients. Okay, so eventually we would have water here. Okay, this is water. And we have because they can pass through the tiniest holes, they are the smallest here, and the inorganic salts are here. Okay, so I'm trying to show you Na and Cl, okay, the stable salt, like inorganic salt, and we have here the lipids, So let's draw them in, lipids are usually yellow, so let's draw them like this. So salt, and this is fats, and lipids and so on. Proteins, and here we have the viruses, okay? Viruses, let's do them in green. Okay, so the viruses are here. Okay, virus. I don't know how to draw the virus, but it's like it has legs and also like a trunk that it can penetrate the cell. So these are the viruses. And we want to keep everything except this, okay? So this we destroy with the radiation, okay? And the rest we keep, and this is the healthy blood of the mosquito. Okay, so then you have things which are a lot bigger. Okay, we talked about it. These are these. Here it's painted like that. And after you rotate it in the centrifuge or something, then you have this is this one. You've seen, you understood. Okay. So what we need is a series of chambers. Between the chambers we pass the liquid through a semi-permeable membrane. Okay, these are the filters. But with each chamber that we proceed, the holes in the membrane are smaller. Okay, so here big, smaller, 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 smaller holes. These are tiny, tiny holes that only the water can penetrate through all of this. And only the salts can penetrate until here. And only the lipids can penetrate until here, and so on. Okay, so the first chamber catches the biggest component, and then the others go through. Catches the biggest, and the others go through. Then the next chamber catches the second biggest and let the others go through, and so on. If it's better, we can use centrifuges for the same thing, and this will order the components by weight. Once we have all the components separated, we destroy the virus, for example, with a blast of energy, of high energy radiation. Okay, not this one, but we take the same technology and shoot it here. And then we combine the components back together, all these components except the virus, and inject it back to the insect's body. Okay, now passive saliva sterilizer using electricity from solar power and battery. We need a tiny amount of nuclear waste. Okay, so this is how another way to do this. Okay, where to get the radiation without electricity. So we need a tiny So this is like another model of the same thing. Passive saliva sterilizer using electricity from solar power and battery. So, well, I don't know why I wrote using electricity, because this one is without electricity. That's the whole point. So the passive saliva sterilizer. We need a tiny amount of nuclear waste. Okay, so this is one model, and now we'll see another model. So in that model we have a tiny amount of nuclear waste. Let's draw it in light brown. Okay, so this is or maybe always like with the green Hulk, the gamma is like in green. So let's do it in green. Okay, so light green. So we have a nuclear waste. Can you see what's written in light green? No. Okay. So let's do this in darker green. Let's erase a little. Okay. So passive saliva sterilizer. Of course, the saliva is the mosquito's saliva. Okay, sterilizer. Okay, so we have the mosquito again. And no, it's not like this, it's like that. And here is the head of the mosquito. Okay, and okay, sorry for not being too gifted in drawing, but this is like the mosquito. And we need a tiny amount of nuclear waste. Okay, so like from a nuclear plant or what's left of a nuclear bomb that you didn't use. nuclear waste. Okay, tiny amount. This is our nuclear waste, like the types used in medicine, which are gamma ray emitters. Okay, this gives off gamma rays. And this is particles, radiation. Okay, so we use them in, you can see in Wikipedia, radioactive waste medicine. Okay, hashtag medicine in Wikipedia. Radioactive waste medicine. Hashtag medicine. Okay, nuclear waste doesn't cost anything. The problem is the opposite, what to do with it. Okay, so here is what we do with it. Since we are talking about tiny amounts, there is no risk of someone collecting the bags and building a dirty bomb. Okay, because it's such a tiny amount. They have to go through all of Africa to get a few grams. Now we need a canister, like a tin can. Okay, but made of lead. Okay, so we have a canister. Okay, this is our canister made of lead. Lead canister. Why lead? Because it stops the radiation. If you put this in here, then the radiation is no longer dangerous. So let's put it in here. Okay, this is the nuclear waste. So now it's inside the canister, the lead canister, like a tin can. And but made of lead. That's because lead blocks radiation, and we want the genitals of the mosquito not to be radiated, only their mouth. Because if we radiate the mosquito's reproductive system, it cannot reproduce. So it's not good for us. So we want it to reproduce, so we need to protect the mosquito. We want to radiate it to here, but not to here. Okay. The top of the canister we close with a lid, which is also made of lead. It's like the lid of a cooking pot. Okay, so there is a lid here. Okay, like a cooking pot. And this is also from lead. Okay, lead. It's like a lid of a cooking pot, but made of lead, and matches the size of our canister. We also drill small holes into the canister, 2 holes close to the top, so that we can tie a string, so that we can hang the canister from a branch of a tree so that ants cannot reach it, but flying mosquitoes can. Okay, so this whole thing is on a tree. I don't want to draw the tree, it's 45 minutes already, but here there is a branch of a tree, and this whole thing hangs from a tree. We tie the tiny amount of radioactive material to the lid from the inside, for example inside a small plastic bag that is tied with a little string to the lid. We drill a small hole in the lid for this. So, now we drill tiny holes all around, close to the bottom of the canister. The holes should be small enough so that when we pour water with sugar into the canister, the water doesn't drip, but big enough so that a mosquito can put its drinking straw, okay, and suck sugary water. Okay, so here we have small holes all around the small hole, and here we put sugary water. Okay, so the mosquito comes to drink the sugary water, puts its trunk, its this thing, I don't remember the name, into the hole and gets radiated. Okay, the water doesn't block the radiation. If we block it in space, it's like something else, we have a lot of water. Here there is a tiny amount of water, it's very close, the water doesn't help. Okay. So, it uses, it puts in the drinking straw and sucks sugary water. When the mosquito puts its drinking straw into the tiny hole, his mouth is exposed to the radiation that cleans his saliva from viruses. Because the gamma rays mutate the virus, and the virus is too simple to have correction mechanism, unlike a cell. So the mosquito is not damaged, the virus is damaged. Every once in a while, the people in the village take it down, this whole thing, open the lid, pour some more syrup, which is just sugar and water boiled together. Okay, syrup. I don't know, maybe it's with you. Okay, maybe it's like this, I don't know. So, which is just sugar boiled and water boiled together into this feeding and sterilizing station. The tiny amount of exposure to the people, very little material over a short time, doesn't do any damage to a creature the size of a human. Okay. Remember, this is very, very small. Okay, now we go to active saliva sterilizer. and then we don't need the nuclear waste. Using electricity from solar power and battery. Okay, this is active, so it's using electricity. Now it is, that the first one was a mistake. Now we are using electricity from solar power and battery. Okay, we do something similar but without radioactive material. Instead we make X-rays using electricity. We make X-rays using a Crookes tube. Okay, Crookes tube. Sorry. Crook like this is something else. Is a villain. Okay, so we are doing like Crookes. Crookes tube. Okay, so a Crookes tube, I'm not explaining, it's something like this, and it has one electrode shaped like this, and that they saw, and the electrode is here, and it shoots electrons, and it gives X-rays. Okay, so that's how Röntgen discovered the X-rays. Okay, so what time is it? 53 minutes almost, okay. But the idea is the same. We use lead to stop the X-rays from harming the mosquito's reproductive system. So this is here, and it has electricity from the solar panels or the battery at night, and it gives X-rays inside here. So I'm not drawing everything, I'm just drawing the X-rays. Like here we have X-rays. So here we have X-rays. X-rays. Okay. And again, these X-rays radiate the mosquito's mouth and kills the virus. But the idea is the same. We use lead to stop the X-rays from harming the mosquito's reproductive system, and the short blast of X-rays sterilizes only the mosquito's mouth. Okay, now continuing with the musical, the second character answers. Miss Guan, laughing. I'm sorry, I just can't stop thinking of Spider-Man that got his abilities after a bite from a radioactive spider. Mr. Lau, laughing. I created a monster, Mosquito Man. I guess with great power there must also come great responsibility. They both laugh. Miss Guan, you always have good ideas. Okay, now mosquito microdialysis. So here I'm rushing through this because I want to have time to finish. So excuse me for being rushed here. We do something similar. I'm sorry. Mosquito microdialysis. Okay, so here I'm really rushing, I'm sorry. It's 56 minutes and I already explained it like really thoroughly, the microdialysis. So now it will be explained again in a hurry. We do something similar, I'm sorry, sorry, mosquito microdialysis. So what about the other method? Ms. Guan asks him. The dialysis of the mosquito's blood. That one sounds more complicated. Is it possible? Mr. Lau. Now Mr. Lau answers, I think it's possible. We have the technique of microdialysis, for example, for brain tissue. Okay, you can see in Wikipedia, microdialysis. And Japanese scientists already used it on tobacco cutworm, which is a moth. Okay, a flying moth, like a butterfly. I'm giving you the link in the description, okay? So it's Yuji Ikemoto, Satoru Kawai, and Junya Mizutani, 1993, microdialysis for the analysis of insect hemolymph in bioscience, biotechnology, and biochemistry. Okay, I'll give you the link in the description. And here is another study that uses it on Blatella germanica, which is a cockroach. A microdialysis study of alatostatin degradation in Blatella germanica dictyotepetra. Sorry, I can't read it fast. You'll see it in the description. This is by Enrique Peralta, Luisa Villaplana, Nuria Pascal, Cristina Carreno, Maria Dolores Piolaches, David Andrew, and Xavier Beles from Physiological Entomology. Okay, so it's in the links. They even have a photo of the cockroach with the microdialysis. So I'm pretty sure we can do it with the mosquito. We don't have time to pump out all the fluids from one mosquito and then clean them properly and then pump them back into the mosquito. So what we should do is sacrifice the first mosquito, which dies, but buys us time. We take the fluids from the first mosquito and then clean them properly and store them. And then, when the second mosquito comes, we have a clean blood donation waiting for him. And the second mosquito's blood will be used for the third mosquito, and so on. If the volume of the mosquito's fluids is too small for doing such a process, remember the chambers that we did in the centrifuge. Remember, with the holes, bigger and bigger, smaller and smaller holes. So if this process needs more fluid, if the volumes of the mosquito's fluids is too small for doing such processes like filtering through membranes or separating with centrifuges, the machine can dilute it with distilled water after we pump the fluid out of the mosquito, and filter out that same volume of water before we pump the fluid back into the next mosquito. Okay, how do we pin down the insect without hurting it so we can later release it unharmed? For this, we can use the centrifugal force that water in a bucket experience when you swing the bucket around and the water is glued to the bucket and doesn't spill like they would normally do. Okay, if the bucket is upside down, the water doesn't spill because we rotate it. It sticks to the bucket, so the same way the mosquito will be sticked to what we're about to do. So this is in Wikipedia, centrifugal force. 1 hour, okay. So we need to really hurry. The machine will have a small room with an open door. The machine puts a small drop of syrup as bait to come in. When the insect is inside, the door closes. The insect finishes drinking the syrup. Then the whole room has a very strong gravity, so the insect cannot fly and is flat on the floor of the room. In reality, what happens is that the tiny room is installed on a rotating arm. And after the door closes and the insect finishes drinking, okay, like this is the room and this is the arm. And after the door closed and the insect finished drinking, the rotating arm starts to rotate the room faster and faster. But the insect only feels strong gravity that glues him to the floor without causing him any harm. Inside the room, on the ceiling of the room, there is a tiny camera so that the computer can see where the insect is. The floor of the room has tiny holes drilled into it in regular intervals. Imagine a chessboard where in every corner of every square there is a tiny hole. From the other side of the floor, I mean below the floor and outside of the room, we install a small robotic needle which we inject into the insect through one of the tiny holes. Using the camera above the insect, which tells the computer which hole to pick. Like a 3D printer where the nozzle moves around the X and Y axis until it reaches the exact point and then it injects the plastic resin, so like a 3D printer has this movement. But here we don't have a nozzle, instead we have a needle. A more sophisticated design would have higher resolution chessboard and two needles, one that can pump out the dirty liquids from one end of the insect and another that at the same time pumps in the clean liquid into the other side of the insect. This better design is also healthier for the insect. At the end of the process, we pull the needles out of the insect. We slow down the rotation gradually, and we open the door so it can fly free. And we put a drop of syrup for the next insect to come. Miss Guan, laughing again. I'm sorry, now I just can't stop thinking about Indiana Jones and the Temple of Doom. It's like you combined the bug tunnel and the death trap with the spikes. Mr. Lau, laughing. There are 2 dead people down here. There are gonna be 2 dead people in here, Harry. They both laugh. Miss Guan, but seriously now, I really think it can save many lives. So, I'm sorry. Can someone produce it in a reasonable enough price so that the rich global north will buy many of these machines for the poor global south? And with the climate change, this blow will hit rich countries too. Mr. Lau, thank you. I also hope it works and that it will be put into good use. But I'm getting back now to where we started. As the saying goes, we shouldn't use a sledgehammer to crack a nut. If I can invent 2 methods to solve this, then scientists can use these methods or find better methods and solve this without genetics. Okay, so that was my invention. So I hope it was clear, and I'll see you in my next invention. And thank you very much. Thank you.